If you're in the environmental health field, you already know that just measuring total PFOS doesn't give us the whole picture. Well, a fantastic new paper from Kang and colleagues in Environmental Science and Technology is really pushing us to look deeper. It gets right to the heart of a critical question. What actually determines the specific mix of PFOS isomers floating around in our bodies?
All right, so here's our game plan. First, we're going to lay out the core isomer question. Then we'll dive into the evidence following two amazing storylines the paper uncovers. One is the exposure story, which is all about our diet, and the other is the excretion story, which is what's happening inside our own bodies. After that, we'll look at some really surprising demographic findings, and we'll wrap it all up with what this means for the future of our research.
Okay, let's start by framing the central issue here.
Right, so as we all know, the legacy PFOS made from processes like electrochemical fluorination, well, it wasn't just one single molecule, was it? No, it was this really consistent mix, about 70% linear and 30% branched isomers. You can almost think of that 70-30 split as the original fingerprint from those historical sources.
But here's the thing that gets really interesting. The ratio we actually find in our bodies isn't always 70-30. So the big question Kang and colleagues wanted to answer is why? What's shifting this ratio? Is it our diet? Is it something our bodies are doing? To get to the bottom of this, they leverage data from two incredibly powerful populations, the big national NHANES survey and the multi-ethnic SWAN cohort.
So let's jump into the first piece of this puzzle, our exposure. And the paper builds this really compelling case that it all starts with what we're eating.
You know, I love a good graphical abstract, and this one perfectly captures the central tension they found. It's like this tug-of-war happening in our bodies. You can see on one side, you've got things like eating fish, which seems to pull our body's profile towards having more of that linear PFOS. But on the other side, you've got things like processed foods and even how well our kidneys work, which seem to pull it back towards the branched isomers.
Okay, so that's the big picture model. Now let's see what the numbers actually showed.
And wow, the data really backs this up beautifully. Just look at the consistency with fish and shellfish. In two totally different populations, eating more fish is linked to a higher percentage of the linear isomer. Now flip that and look at the processed foods, pizza, salty snacks. It's the exact opposite. They're linked with a lower percentage of linear PFOS, which of course means a relatively higher proportion of branched isomers. It's a crystal clear story of dietary divergence.
So what's the mechanism? Well, the paper points to a lot of existing research showing that linear PFOS is just plain better at bioaccumulating up the food web, especially in fish. What that means is, when we eat fish, we're eating something that's already sort of preloaded with a higher proportion of that linear isomer.
But it's not just about what we take in. The paper then pivots to this fascinating story about how our own bodies are actively shaping this isomer ratio through different excretion pathways.
And the leading hypothesis really boils down to this concept of isomer-specific protein binding. The idea is pretty straightforward. Linear PFOS seems to bind more tightly to proteins in our blood, like serum albumin, and because it's holding on for dear life, it's less free to be filtered out by our kidneys, so it effectively becomes harder for our bodies to get rid of. And the study found two really elegant lines of evidence that support this binding idea. First, they saw that premenopausal women, who have an extra way of getting rid of PFOS through menstruation, actually had a higher proportion of linear PFOS. This suggests that their bodies are selectively getting rid of the more weakly bound branched isomers. Then, they saw that people with better kidney function, a higher eGFR, also held on to more linear PFOS. Again, it fits the model perfectly. The freer branched isomers are just easier for healthy kidneys to filter out.
Okay, now we get to what I think is one of the most compelling and honestly most important findings in the whole paper. And that's this incredibly consistent pattern of isomer differences across racial and ethnic groups.
I mean, just look at this data. It is absolutely striking. You've got NHANES on the left and SWAN-MPS on the right. These studies were done a decade apart in different groups of people, but they show the exact same pattern. Non-Hispanic Black and Asian participants have a significantly higher percentage of linear PFOS in their blood compared to non-Hispanic white participants.
And the power of this finding is just in its robustness. This isn't some statistical fluke in one data set. When you see the same exact pattern replicated across different study designs, different decades, different locations, that tells you that this is a real, persistent signal that we absolutely have to pay attention to.
But here's the real kicker. These differences don't go away.
even after they adjust for all the things we just talked about. Diet, kidney function, you name it. This is a massive clue that we're still missing a piece of the puzzle. It strongly suggests that other factors are driving this. Maybe it's genetic differences in protein binding, maybe it's other dietary patterns we haven't measured. It's pointing to a critical and, honestly, exciting new direction for research. So let's bring it all home. What does this all mean for us, as researchers and practitioners working on PFAS?
The biggest take-home message here is that isomer profiles are not just random noise. They are a rich, informative fingerprint of our unique exposures and our unique physiology. This paper makes such a powerful case that just measuring total PFOS really isn't good enough anymore. If we want our work to move forward, we need isomer-specific data. It's what will allow us to ask much more precise questions, like, hey, are the health effects we've been linking to total PFOS actually being driven by one specific isomer?
And that leaves us with this final, crucial question for the future. This work really pushes us to think about how a deeper understanding of isomer-specific toxicology and kinetics could totally change and dramatically improve our risk assessments. It's a clear call to action for the whole field, and just a beautiful example of how getting into the weeds of analytical chemistry can open up brand new avenues in environmental health science.